Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 169th seminar, and I call it a Technical University of Denmark Day. I've just got back from my trip to the SIMB annual conference to give a talk, meet colleagues and friends, and also celebrate its 75th anniversary. I hope SIMB, or the Society for Industrial Microbiology and Biotechnology, will continue to serve as an avenue for researchers. Uh, Professor Irina Borodina is another speaker for today, but due to some company rules, she cannot have her talk recorded. But I just wanted to briefly introduce her because she basically making real impact on the society by basically translating the metabolic engineering technology she developed into real product, worldwide. And so she's a real pioneer in terms of those kind of real transition from the technology development stage into real market translation. She is a professor at the Technical University of Denmark, or the TU. This summer, thanks to her and the organizing committee, we all had a great time in Copenhagen to discuss the past, present, and future of metabolic engineering. She and I also co-authored a perspective article entitled Impacting Future Generations of Synthetic Biologists by Ensuring Diversity, Equity, and Inclusion, published in Trend in Biotechnology, one of the cell journals. And actually the Matt, in the chief editor of the journal, actually kindly invite us to put this wonderful article together. So, Irina, as I mentioned, cannot share also the talk recorded because of the company rules. I'm gonna go to our main speaker of today. So, our main speaker of today is Dr. Wes Robertson. He is from the United States originally, where he grew up mostly in Tennessee. After receiving a bachelor's degree at Vanderbilt, he then moved up to the north to carry out a PhD at Yale, where he did chemical biology research in the laboratory of Professor Alana Shipparts. Afterward, he moved to the United Kingdom, where he did synthetic biology research as a postdoc with Jason Chin at the Medical Research Council Laboratory of Molecular Biology in Cambridge, UK. There, he learned synthetic genomics, and he was part of the team of genome engineers which assembled the first synthetic recorded genome for E. coli. Now he has recently started his own group in Cambridge at Medical Research Council, the Laboratory of Molecular Biology, where he is applying synthetic biology concepts toward aiding microbiome research. Wes, thanks so much for your time today, and please take it away. It's all yours now. Thank you.
Amazing. Thanks so much for the kind introduction. And I'm really happy to be here today and share a bit of the work that I've done in the past, as you mentioned, as a postdoc in Jason's lab at the LMB here in Cambridge in the UK. It'll be a sort of a general talk about that work and how useful total genome syntheses can be to provide new functionalities to microorganisms. And I'll also tell you a little bit about the end of what I'm up to in the lab, which I just started here, also at the LMB. We're a small team right now, but we're up to a few cool projects in synthetic genomics. And I'll tell you a bit about what's going on there. So my background is in synthetic biology, where nowadays, as DNA engineers, we can control the sequence of every nucleotide within a synthetic genome. And in the microorganisms in which which harbor these synthetic genomes, this can confer new to nature properties to them. For example, we've shown one new to nature property can be the ability of these microorganisms to synthesize unnatural polymers in a DNA programmable fashion. Another new to nature property can be via orthogonal genetics, in which we can confer a lack of viral attack to these microorganisms, so complete viral resistance to bacteria which harbor specifically designed synthetic genomes. And quite simply, I'll tell you really a lot about that type of work today, which I've done in the past and some of it recently. But in my current lab, in the new lab, we aim to take these synthetic biology concepts and apply them towards microbiome research, more specifically, apply them towards gut bacteria, which are present in the gut microbiome, because we simply just want to provide more tools which can potentially benefit researchers to then study the endogenous microbiome using, say, synthetic microorganisms, which can be more genetically isolated, or, for example, at least be more biosecure. So all of our engineering feats in synthetic biology are based upon our understanding of the central dogma in molecular biology, in which we know the flow of information, from DNA to RNA to proteins in cells. And what's key to me, at least, is that it's a powerful paradigm for the programmable synthesis and evolvable synthesis of proteins in cells. Nowadays, we also understand in more detail the mechanisms which control the central dogma, specifically through the canonical genetic code, in which the universal canonical genetic code, almost all organisms on Earth have 64 codons encoding for 20 building blocks for the synthesis of proteins. And in this way, starting in the late 1970s, molecular biologists realized that they could actually control the genetic code in microorganisms such as E. coli to turn them into programmable protein factors. Also, during the 1970s, researchers started to sequence the DNA from different organisms, different genes, and they actually realized that in viruses and humans and in different types of bacteria, they all have the same universal conserved genetic code. It's a common communication language. And taking those two bits of information together, you can then express a DNA transgene, such as for insulin, in a microorganism such as E. coli. And that was really the beginning of the biotechnology industry, based upon this understanding of the genetic code. Jumping forward a few decades to the 21st century, the goal of synthetic biologists is to expand upon this paradigm of DNA programmable synthesis, where our goal is to incorporate additional chemistries, non-canonical or unnatural chemistries, so that we can then use DNA to not just synthesize proteins with new chemistries and potentially new functions, but also to synthesize completely unnatural polymers, reminiscent of what chemists can do in a flask. Except the advantage of using the genetic code in cells is that we can do this in a scalable and biosustainable manner. So this is really sort of the goals of synthetic biology. But to be able to do that using cells in viable organisms, we're going to have to actually re-engineer a fundamental component of life, which is really the genetic code. What we wanted to do, this is around the time of when I joined Jason's lab as a postdoc, the lab had the goal of reassigning the genetic code by reassigning some of the redundant codons. Again, 64 codons, 20 building blocks. The goal is to reassign some of that redundancy to have new meaning for encoding for non-canonical amino acids. Let's say in this example, 3 additional non-canonical amino acids available in this code. And that way, you could then turn cells into programmable chemical factories. And also, you could confer to these cells a different or orthogonal genetic code, which would be a selective communication language, which would allow these cells to focus on the task at hand without outside interference from the environment. And this way, you could then use cells to make new plastic type compounds, for example, or new types of novel drugs, and be quite useful in biomedicine and biomaterials. So these are clear goals of what we wanted to do. But there are also clear obstacles of why we couldn't get there. And at the time, I would say there are really 2 big obstacles, which I'll tell you about today. The big DNA problem and the synthetic DNA design problem. Big DNA, how do you go from many small pieces of synthetic DNA to one large megabase scale size genome? And for the synthetic DNA design problem, how do you confer this new to nature property in the synthetic DNA while maintaining the natural biological function which that DNA imparts to enable the cell to replicate and ultimately to enable the organism to grow properly? So these are 2 big challenges. In terms of the blueprint for how we thought about going about to do this, we called it sense codon reassignment. And I'll briefly take you through this, where you start off with a gray wild type genome operating off a wild type canonical genetic code. And you can take, let's say, 2 target codons for serine, 2 of the 6, and recode them to synonyms, and by that I mean replacing them with synonyms throughout the entire genome in a process we call codon compression. That will allow you to then delete the corresponding tRNAs or transfer RNAs or release factors which used to decode those removed codons. Then you have blank codons, and you can impart a new meaning and reassign a new meaning to blank codons by importing in engineered orthogonal amino-acyl tRNA synthetase, tRNA pairs, which we've taken from previous years of research from many different synthetic biology labs. So in that way, this takes you all the way to reassigning a genetic code in an organism with a synthetic genome. So this is the blueprint. And now I'll guide you more slowly through this process and for what we actually did for the first synthetic genome example. So we wanted to recode the E. coli genome. It's a great model organism. It's well studied, and it's quite useful in molecular biology. And we know that in the canonical genetic code, there's a lot of redundancy. 18 out of the 20 amino acids are encoded by more than one synonym. We asked a simple question. For the serine codon box, there are 6 codons coded by 4 tRNAs for just 1 amino acid. Can we simply go from a 6 to a 4 codon box? Can life survive with this code compression? And to do that, we wanted to systematically replace codons with synonyms, process we call recoding, and overall called codon compression. So in this case, for example, we wanted to replace TCG and TCA codons with any of these 4 synonyms. Then we can delete the associated tRNAs, which used to decode those codons. And then again, we can import in orthogonal translational machineries, which will reassign a new meaning for an unnatural amino acid to this liberated free codon. The thing is, there are tens of thousands of these serine sense codons throughout the E. coli genome. We couldn't just do this through site-directed mutagenesis. That would take a very long time. We wanted to just do this via a de novo genome synthesis. That was the tractable way to go about this. And so tens of thousands of changes you'd have to make. And to address the big DNA problem, a previous postdoc in the lab named Kai-Hung Wang, who now has his own lab at Caltech, he developed a technique called REXer. And this was an approach to replace wild-type DNA on the genome with synthetic DNA at scale. You can import in a bacterial artificial chromosome harboring about 100 kb of synthetic DNA, depicted in pink here. And then you could trigger the activity of Cas9 to linearize this back, and then the activity of the lambda red recombination machinery to then integrate it in its corresponding homologous location in the genome. And along with the judicious use of negative and positive selection markers, you can ensure this process occurs with high efficiency and fidelity. And overall, what Kai and others have shown is that Rexer work quite efficiently in a size-independent manner. And so this was an approach to address the big DNA problem, but what about the synthetic DNA design problem? What about the genome design? If you're going to recode two target codons to any of these four synonyms, there are many end of the P types of potential genome designs. It's an astronomical amount. And so we really wanted to have a tractable approach to address this. And what the lab came up with was an approach where they would have different pieces of synthetic DNA around 20 kb in size, which focused on the DCW cluster, which is about a 20 kb region of E. coli genome rich in essential genes required for cell division and cell wall biosynthesis. And after performing a series of Rexer experiments with different synthetic DNA designs harboring different recoding schemes, in this case, 3 different serine schemes, you could simply do NGS post-Rexer on individual clones and then plot the uptake of synthetic DNA or recoding. And what you can see here is this scheme failed, but these two were just fine. So in the end, scheme 3 was taken, which replaces these 3 target codons, 2 serine, 1 stop, with these 3 synonyms. And then based upon the success of this initial scheme, it was then extrapolated to the rest, the 4 megabases of the synthetic E. coli genome design, which in the end harbored about 18,000 recoding events, which you can see actually plotted here is red lines across this synthetic genome design. So this was the initial design for a recoded genome. And then the challenge was, okay, how are we going to actually boot this up? Going from a computational design to an actual viable bacterial cell. So to do this, we teamed up multiple of us genome engineers in the lab, and we broke up the genome in a retrosynthetic manner. Started off with a 4 megabase synthetic genome design and broke it up into about 8 sections of half a megabase each. And each section was broken up into 5 fragments of 100,000 base pairs each. And each fragment is broken up into 10 stretches of 10 kb each. And it's these stretches of 10 kb you can order from a commercial DNA synthesis vendor. So we had a few of these, and we'd use yeast to go from these individual pieces to 100 kb back yak. And this was really the functional unit we would use to perform our Rexer experiments to test to see how well the synthetic DNA was taken up on the genome in this E. coli strains. And so what's useful about Rexer is you can iterate it. You can alternate between different selection markers, different combinations of antibiotic and toxin cassettes, so that you could perform 1, 2, 3, up to 5 steps in a row quite efficiently, which would afford a semi-synthetic strain of about a half a megabase of synthetic DNA in this strain. And so this is what we did, dividing and conquering in different regions of the genome simultaneously. And I make it sound all easy, but actually we did run into problems. There were synthetic design exceptions. There were problematic regions which were recalcitrant to recoding. And Rexer helped us identify these locations initially, where after plotting a recoding landscape, you can see that in these 2 regions, there are these values, there's no recoding at all. It just maintained the wild type sequence, no synthetic DNA sequence there. And in this case, to overcome this, we realized in this region here, there was a specific refactoring event, which didn't have really much of a synthetic insert in between the 2 ORFs, which were separated to allow you to independently recode ORF1, for example, without simultaneously messing up ORF2. So essentially, our refactoring scheme in this case seemed not to work. But then when we added a larger synthetic insert of around 182 base pairs and put this on a back and did Rexer again, which you can see in this purple line, this purple recoding landscape, that successfully was able to recode that part of the genome and fix this problem. And overall, that's just one example out of 4 problematic positions in this genome design, where it contained a total of 7 idiosyncratic codons. But out of 18,214 recoding events, in the initial design. It's actually quite amazing how efficient the initial design was in this case. But one takeaway for me on this was that fixing takes time. It takes time to locate the position of the problem as well as to fix it with an alternative synthetic DNA design. And this ended up taking quite a bit of time for us. But needless to say, with all these problems fixed in different strains, which harbor different regions of synthetic DNA, in different areas of the genome, we then needed an approach to combine it all together in a synthetic genome assembly approach. So we sort of developed a variation on a conjugative genome assembly approach, which was developed by Farron Isaac's lab originally, in which quite simply to have a donor strain with enough plasmid to donate one region of synthetic DNA harbored on the genome, which is downstream of a separate region of synthetic DNA, which is harbored on the recipient strain. And essentially, transconjugates will increasingly be recoded in a clockwise fashion across the genome wheel. And so finally, after a series of convergent conjugation steps, it yielded a fully synthetic strain, which we called SYN61. And we're quite encouraged. It's alive. This organism lives off 61 codons, quite different than the universal genetic code. Phenotypically, it could look similar to the parental MDS42 E. coli strain, which is the basis of the design. But we did note that it didn't grow as well. It was recoded, had the same proteome, different DNA sequence, obviously, but there was a hit on the growth rate. About 60% of that, of the parental strain. And for me, this is actually an opportunity from this example to focus on this codon choice problem for future genome designs and future genome synthesis. Is something that focusing on now in my lab. But needless to say, we could boost the growth rate up with common adaptive evolution approaches and then explore these new to nature functions which could be imparted by this synthetic genome. And I'll tell you briefly about two: expanded genetic code for non-canonical biopolymer synthesis, as well as orthogonal genetics. But to start with the SYN61 strain, we then deleted the associated tRNAs, SER-U, or release factors, purified gene, which used to decode the removed codons. And this yielded the strain called SYN61 delta 3, which quite simply we asked, what is a consequence of 3 blank codons in this synthetic organism? We and others hypothesized that any foreign genetic material operating off the wild-type genetic code, when coming into the SYN61 delta 3 cells, it would not be able to have its material translated and produced because the ribosome would stall on these sense codons because there are no tRNAs or release factors to code them properly. And so then result is, you won't be able to make these proteins, shouldn't be able to replicate, shouldn't be a problem to have this T6 phage, for example, which has many instances of these sense codons spread throughout its genome. And in fact, Julius Frieden's postdoc at the time showed with T6, so you could, titrate it in, you could detect it initially, and then you could see how it expanded over time post-infection in control cells pre-tRNA knockout. But post-tRNA knockout in the delta 3 strain, it acted the same as metabolically dead control, which is phage comes in and then it just gets diluted away because it can't replicate its own genetic material. And the end result is cells survive and grow robustly in the presence of the phage, whereas cells pre-tRNA knockouts die because the phage causes lysis, etc. So this was initial proof of principle for phage resistance. And then Julius also showed that in a cocktail of multiple different types of phage, in the absence or presence of the phage, the delta 3 cells grew fine. So this is really just one flow of information being blocked. What we call this unidirectional genetic isolation. So this is the flow of wild-type information not coming into our recoded cells. What about the flow of information coming from our recoded cells and escaping out into the environment? What about this bidirectional genetic isolation? And this is a question specifically at Jerome, a PhD student in the lab, focused on initially. And so we asked, can we benefit from the flexibility of SYN61's genetic code to demonstrate this principle of bidirectional genetic isolation, sort of to allow us to better genetically control the flow of information between organisms? And through a series of horizontal gene transfer experiments, you could have a donor gene, for example, for antibiotic resistance gene. And if it's donated into recipient cells, which operate off the wild-type genetic code, TCG and TCA will be decoded as serine. The end result is a correctly translated protein and, for example, antibiotic resistance. Great. But if the donor are recoded Syn61 cells in which we've refactored the genetic code and changed the identity of TCG and TCA to encode for alanine and histidine, which you can do by just simply changing the anticodon of the native alanine and histidyl tRNAs, then this message for this protein is not read properly in wild-type recipient cells because these will be decoded as serine, not alanine or histidine, and the end result is a missynthesized protein with improper function, no antibiotic resistance, for example. So the result is we've aggregated the flow of information from the recoded cell out into the environment. This works vice versa as well. So really, for me, this is a system that's set up that is enabling the control of communication between the synthetic cells and environment and vice versa. And, it's not just a biocontainment strategy at a strain level or genome level, but really it can be applied at the gene level. Which I think will be quite useful in future research, both in the academic labs and potentially industrial labs as well. So that was orthogonal genetics. What about expanded genetic code? Quite simply, we had 3 open codons, and we showed that we could reassign the meaning of these after importing in mutually orthogonal synthetase tRNA pairs, which could then decode these 3 open codons with 3 distinct non-canonical amino acids. And in one reporter protein, in this case ubiquitin with a C-terminal his tag, I could do a Western blot and show that only in the presence of all 3 non-canonical amino acids would we get full-length ubiquitin expression and thus detection. And it was synthesized at wild-type levels, and it occurred with quite high fidelity, as we could tell and judge by mass spectrometry. But our goal wasn't just proteins decorated with unnatural chemistries. Our goal was completely non-canonical biopolymers translated by the genetic code in cells. And so to do this, initial proof of principles were set up where I just had an N-terminal GFP reporter with a string of liberated TCG and TAG codons. And we could make these type of polymers, N-terminals of GFP, and detect fluorescence. And indeed, we could just simply transform in different combinations of synthetase and tRNA pairs and spike in different combinations of corresponding non-canonical monomers to then change the reassignment schemes to then have quite different encoded sequences or polymers, which again works quite well with high fidelity. And then Martin Spink, a postdoc in the lab at the time, took this to another level and really developed a system that you could make cyclic peptides in a programmable way, where naturally these can be found as natural products. They can be antibiotics or immunosuppressants, for example, but in nature, they usually use large megadalton enzymatic enzyme complexes to make these, not really highly amenable to programming in the same way that DNA is. If different DNA input, different combination of synthetase and tRNA pairs, and you can get a diverse amount of different macrocycles and even DEPC peptides, which are macrocycles containing an ester linkage in their backbone. And specifically, Carlos did a good job of showing how the pyrrolysine synthetase tRNA pair can accommodate hydroxy acids, not just amino acids, to thus allow you to add an ester linkage to these cyclic peptides. And overall, we showed that you could have quite a diversity of different types of macrocycles which are made in cells in a DNA programmable manner. And this can be set up for screens or selections by others if they're interested to fish out specific activities or binding abilities, etc. It was around this time that I was finishing my postdoc and looking to set up my own lab. My sort of outlook on synthetic genomics was in terms of a timeline of DNA synthesis. There have really only been 2 examples of bacterial genomes. Craig Venter, Jen Gibson, 2010, Mycoplasma, 1 megabase, Syn61, 4 megabase, I just mentioned, but actually we just finished Syn57, which is a recoded E. coli genome with a 57 codon genetic code, where we removed 7 codons throughout the entire genome to yield, to our knowledge, the most codon compressed living organism that exists, and really credit to a strong team of genome engineers in the lab, including Fabian, Martin, Rothy, and Rongzhen, in Jason's lab, where we finished this together. And we were quite encouraged that life could survive based upon just 57 codons, even in a genome which harbored over 100,000 recoding events. And really key to this were innovations which the team developed for fixing problematic regions where recoding or synthetic DNA design could decrease the fitness of the organism. But at each stage, through sort of mapping and fixing these recoding problems which led to fitness defects, we could then improve the fitness of the semi-synthetic strains and then assemble them all together to yield one final fully recoded synthetic strain, which we call SYN57. And this would be quite useful for the same applications which I mentioned earlier for SYN61. It's of the benefit here is you have 7 open codons, which in future work can be shown through codon reassignment experiments to potentially incorporate up to 7 non-canonical monomers at once in one message. And again, credit to a strong team here for completing this project. And this paper, actually, you should check it out. It's going to come out in a few hours. So in terms of my outlook on synthetic genomics, we only have 3 examples, but really genomics can redefine how organisms interact with each other and what organisms can produce. I think this can be quite useful in academic and industrial research. And for me in my lab, what we want to do is we want to improve the tools for synthetic genome design and synthesis. And we also want to move beyond model organisms like E. coli and mycoplasma. We want to move to more interesting organisms which might be significant for human health. For example, gut bacteria and the microbiome. Quite simply, we programmed the model, E. coli, in the lab, in the flask, and now we want to focus on developing synthetic genomic tools so that we can actually recode and make from scratch the genomes for gut bacteria, which can then be deployed in a biosecure manner to aid in microbiome research. So to do that, currently a postdoc in the lab, Gonzalo, is working on setting up a system for large-scale synthetic DNA integration in the gut bacteria, initially thinking about some Bacteroides type species. And first, he's demonstrating that he can deliver synthetic DNA into the gut bacteria. And then in the bacteria harboring the synthetic DNA, he's showing that he can do recombination of the synthetic DNA to swap out the corresponding wild-type region in a similar manner that we've shown before, but using different tools and a slightly different approach to what was done before in E. coli. So quite excited to see sort of Gonzalo's progress on this. And this would be like a necessary technology development to enable the total synthesis of a different type of gut bacteria. And we think this would be quite useful because there's some, for us, quite salient reasons why recoded cells could be advantageous, not just in the microbiome, but in general for biotherapeutics, where you can not have horizontal gene transfer, you can not have problems, genetic instability due to transposons, for example. Moreover, if you're going to make an entire genome from scratch, you can design in large-scale native bacterial features made from other species and make more types of chimeric type synthetic genomes, for example, to give privileged access to nutrients such as certain types of sugars in the gut. This could be quite useful. And at least it could provide an approach to biosecure these bacteria, which could be deployed as biotherapeutics. Again, not just biocontaining them at the strain level or genome level, but you could biocontain at the individual gene level through these approaches of bidirectional genetic isolation I mentioned earlier. Yeah, and we think that this would be quite useful because actually others have shown that in the human gut, through longitudinal studies, they could see the acquisition of, depending on the species, over hundreds of genes per year. So in terms of long-term stability, horizontal gene transfer is really changing these species quite more than I initially thought before reading into this. And also not just gaining genes, which could turn, for example, biotherapeutic into a pathogen, which you don't want. Genes are also lost at a similar rate. For example, if you have a biotherapeutic, which has a specific synthetic gene circuit to detect inflammation, for example, that could be lost due to sort of horizontal gene transfer based mechanisms. So we're hoping that, the sort of recoding approach can address some of these genetic stability problems. And quite simply, we're also we know it's great, but there's an impaired fitness problem. So we're addressing this as well. In a complex environment, competing for resources with other bacteria, we need to have high fitness levels. Otherwise, it's just not going to survive. It's not going to be useful. So we're addressing this by trying to better design genomes which accommodate recoding and fitness. And Tridi Rao, PhD student of that, is focusing on this based upon leveraging previous research in DNA-based large language models, which really can enable prediction generation and some type of understanding of how DNA operates at the genomic level. And these are often trained on thousands of prokaryotic genomes from open databases. And quite simply, we just want to use them to better understand codon choice in our synthetic organisms. And 3D wants to use them not necessarily just to generate from scratch a synthetic genome design with this algorithm, but at least help us pinpoint design flaws before we actually order synthetic DNA and before we find it's a problem for fitness later on during the genome engineering project. We think this will be quite useful as well as save a lot of time for us and others when making a genome from scratch. And with that, I'd just sort of like to wrap up and sort of mention sort of the lab's goals, designing different types of biotherapeutic or native bacteria, imparting different features of recoding into their genomes, with the goal quite simply to show in mouse models that we can have recoded biosecure bacteria, which could have advantageous features such as you could then selectively communicate with them through selective horizontal gene transfer experiments. For example, conjugating a specific message, which is only read by your synthetic bacteria and not read, doesn't affect the endogenous microbiome. So with that, I really like to thank you for the time and shout out to the team. And we have two new students, Angus and Rodrigue, joining in the fall. Thanks a lot to Jason and past members of his lab. Worked a lot with them in these two different genome engineering projects. And happy to take any questions.
Fantastic talk. I really love it. I want to
Thanks.
check with Irina whether she has question or comment, or she is out. Okay, let me start. So, I have multiple questions, but let me start with a simple one. So,
Sure.
I believe the genetic, codon 64, that is the universal across the different kingdom of life, right?
Yeah.
So the question is, so for example, you kind of manipulate the setting codons, six codons, and I just
Yeah.
wondering why that specific amino acid has six codons. Is it proportional to total abundance in the organism, or how that, that kind of evolve? I mean, not, the only thing I, believe in biology is evolution or something else. I mean, but the largest contribution or largest factor is, I believe, evolution. I believe the what we are right now is because of evolution. But why we have six codons for serine but not for other amino acid? That's my question.
Yeah. I mean, this is sort of an origin of life research question, which is a topic in its own right, which I think is quite conceptually intriguing. Um, we don't have a lot of answers for that. And one reason is because of the frozen accident hypothesis, which Crick set forward initially in 1968, which is that, yeah, essentially in the beginning, essentially billions of years ago, when life started, and started to replicate and store information through mechanisms like genetic code, it just arrived upon this solution all of a sudden of this universal genetic code, and then boom, life ran off with it. And that was a system that started from the beginning. And we don't really have any evidence necessarily through, you'd think, oh, it'd be nice to look at some in terms of fossil record type analogies, like some intermediate genetic codes to see how we arrived upon the 64 one. But we don't have that. Again, it was a frozen accident. Boom, it worked, and then life ran off with it. And since horizontal gene transfer is a big mechanism of evolution, it was advantageous for all organisms, especially starting off with, cells and protocells or whatever, and microorganisms to share genetic material because this is advantageous over long amounts of time for them to evolve and survive and thus be fit in terms of reproduction. Yeah, so to get more specifically to your question of why six codons, I don't know.
Mm-hmm.
I don't think it corresponds to like the abundance of serine, for example.
Mm-hmm.
That's not true. I mean, for example, we know in Syn57, you know, there we recoded four different serine codons to take that from a six codon to a two codon box. But we also recoded alanine codon box. We took that from a four codon box to a two codon box. And they're by far, they're a few fold more alanine codons, even though we only recoded two of them compared to the four of serine, than the serine ones. So alanine is a more abundant residue, is what I'm saying, but it only has a four codon box. So there's no really sort of correlation there in terms of abundance of the residue with number of choices in terms of synonymous codons. I mean, we know the cell can use synonymous codon choice is useful for the cell. It gives it different options to adapt, so it adopts different secondary structures, which might be to encode the same proteome, which might be useful for the regulation of genes in terms
Mm-hmm.
of to dim down or turn up protein expression levels. So this is one reason why it's useful. But in terms of your initial question of why did it start off like that, six codons, why is it like that now? I don't know. It
Okay,
could
so
have just been
that's
random
helpful.
that
That means, I mean, six codon is not because of the abundance of the amino acid.
yeah. Yeah. But what's interesting is like, there are what's interesting is now through total genome synthesis, we can start, I mean, we have started, we're testing these hypotheses. Is six codons required for life for serine? The answer is no. You can either have four or two, actually, now that we've shown with SYN57. So maybe through genome syntheses, we can start to ask some of these questions, um, maybe over some adaptive evolution type, like Linsky style experiments of, do cells prefer this or that, right? In terms of the number of codons. We might not be able to have a clear cut mechanistic explanation for it. But you could at least say, well, this one survives better than that one. So that could be an interesting type setup for those that are interested in sort of genetic code evolution.
I see. I see. Thank you. Yeah. So because I mean the kind of thinking about this kind of problem whenever I see, this type of paper, the question was why we have 20 amino acid only, not I mean we have the some rare amino acid that also, you use, but why
Yeah.
not? I mean 19, why not 21? I mean, that's kind of my question I always kind of
Yeah. I mean,
wanted to
it's pros
know.
and accidents. That's the answer.
So that
It's maybe
means
not very satisfying, but that's the current hypothesis. Yeah.
So if that is an accident, that means we are not at the global optimum in terms of evolution. That is somehow promising because if you find some recorded, organism, we may have some better at the end of the strain at the end of the day. I mean, if that is the case, right?
Yeah. Yeah, it's very much possible. I mean, the idea is that over long-term evolutionary timelines, it's disadvantageous to dramatically change the genetic code. It would take a long time. And it would be disadvantageous, for example, because you're missing out on certain benefits of horizontal gene transfer, right? So, that's why it hasn't emerged in nature yet. But now through genome synthesis, we can just more quickly make it and test these hypotheses, sort of insulated from the whole evolutionary pressures and the long-term evolutionary pressures, right? And then, as you say, we could then test and see, like, actually, can we find a better optimum now? Like, are these microorganisms better off in surviving in certain environments if they have, either a compressed genetic code or, for example, um, additional building blocks available for their sort of chemical repertoire to make their proteins, right?
Mm-hmm.
So those are interesting questions. Yeah.
Sure. Let me check again. Irina, are you there? Okay.
Oh, I think she's back.
Oh, do you have any comment or question?
I'm here. Well, first of all, it was very nice talk. Thank you so much. That was
Oh, thanks.
extremely interesting. I've been also following the yeast synthetic genomics project, where they were also trying to where they're also trying to recode, and actually don't know how many chromosomes they have gotten up to, but I think they also replaced one of the stop codons,
Yeah,
so that
yeah,
they
yeah.
transferred one of the stop codons. So do you know why it's so much more difficult, or maybe they just didn't plan it in to replace more than just one in an
Well,
eukaryote?
yeah, I mean, I think the Yeast 2.0 sort of consortium is quite interesting. A lot of people involved been going at least since 2011 or so. And I think to answer your question, they've made all the individual chromosomes, but the problem is combining them all together. They're still working on debugging essentially synthetically lethal effects, but they can't combine these two genotypes together in a viable single organism. And yes, they've recoded the amber codon in these designs. I think in 2023 they had a paper where they had about just over 50% um, of the chromosomes combined in one strain, and so they're working on improving the fitness of that strain and adding the rest. But to answer your question of why is it more difficult, whatever, I mean, yes. Eukaryotic systems are more complex. The changes they made, not just the recoding change, the single codon they looked at, but also they add a bunch of LOXP sites. They rearranged the location of many of the, if not all of the tRNAs. These are interesting experiments, but they're all synthetic genome design changes. And all of these changes had an effect which they couldn't predict in terms of affecting the fitness of the organism once they tried to combine all the synthetic chromosomes at once. And so quite simply, too many changes can take a longer amount of time to debug, map out the location of the defects, and then fix them. And that's at least my hypothesis for why it's not finished yet. But I do hope it'll be finished one day. It'd be nice to add as the fourth synthetic microorganism, with synthetic genome, right? And like obviously yeast have many other cool applications that, bacteria can't do. So it'd be interesting to see, what their plans are for that.
I was also wondering about the potential approval registration of your synthetic E. coli cells for different applications, both therapeutical and also agricultural, particularly when it comes to life bacteria applications. And they are extremely heavily engineered. So do you think there is any prospects there of getting such organisms registered?
Yeah, I think there are. I mean, my goal as an academic is not, to get in the clinic right now, right? But at some point to make these strains available so that others, can think about it if they want. And the way I usually describe this to people is that these experiments are already happening. These clinical trials are already happening, obviously, with wild type based biotherapeutics. Some of them still have like colobactin toxin, gene like operons in them still, which are, toxic to individuals, things like this. The point is like through a genome synthesis, you can better control every aspect of it to more so safeguard it compared to what's already being done with wild type biotherapeutics. So if you ask me, it's sort of more responsible way to do this. And I would, that's sort of how I would try to describe it to somebody that would be a skeptic of GMOs or something like this. Although in ways it is hard to predict sometimes how you have to sort of counteract this type of skepticism. I'm sure that you're sort of an expert on this compared to me. Based upon your experiences. But yeah, I think that's my main message is that we can actually control every aspect of it to make it safe, as well as to add kill switches in different biocontainment strategies to immediately kill it if we want to and get rid of it, right? At the organismal level, as well as, again, like, safeguard at the gene level, which is something which is not as necessarily robustly you're able to do with wild type organisms.
Yeah, thank you.
Yeah, but I'm not getting in the clinic anytime soon. So, but yeah.
So, Wes, I have some follow-up question. You mentioned we have total 4 species that basically having synthetic DNA. I believe you are including the Craig Venter's uh, synthetic cell as well. And what is the other one other than yeast and E. coli?
Well, so what I mentioned is that just to Irina is that the fourth synthetic genome would be the yeast 2.0.
Oh, I
If
see, I see, I see, I see.
if that's finished, I don't know, tomorrow or something, right? So that would be the fourth. Yeah.
So
And the
E.
other three,
coli and then the Craig Venter's Mycoplasma. What is the other one? That's my question actually.
well, that's what I'm saying is like there's Craig Venter and then there's Sin61 and Sin57. So those are the
Oh,
three.
I see. Collect two
Yeah, those are the
try
three.
one.
Yeah, yeah. I mean, so also Craig Venter and Clyde Hutchison, the Venter Institute in 2016, they made a minimized version even
Mm-hmm.
of the initial mycoplasma synthetic genome. So they went from 1 to half a megabase, and that
Right,
was very obvious. I mean, you're probably very familiar with this,
right.
knowing John Glass and everybody being at the Venture Institute. That was very cool. I mean, that could be considered another one, but really it was a minimization of the original one.
Right.
So I don't know if that's a new one or not. I don't know what you want to call that, but if we could just consider that as one, and then a minimized version of the same one, then yeah, that's what I would say.
But based on my memory, that strain does not have changed codon, right?
It does not. So they were focusing more on compressing the genome and
Right.
seeing the minimal sort of genetic requirements for life, which is a really cool conceptual question. Whereas we were focused on, compressing the genetic code, not the
Mm-hmm.
genome. So they didn't do any genome-wide recoding.
Right.
Yeah,
So in that
which is
case,
fine. I mean, they were developing a lot of these, sort of instrumental techniques, right, to assemble synthetic DNA, and that was their initial goal, was to show they could do that, right? So,
that I just wanted to make sure, so that's actually the question I have is why not community building recorded other bacterial species? Because yeast has a lot of problem you mentioned because yeast has multiple chromosome, but other bacteria only have one chromosome. You could
yeah.
still do the recording like you did for E. coli, but what is the real challenge for other bacteria?
Yeah, I mean, that's sort of something I'm thinking about all the time now, right? Because we're thinking about gut bacteria, different types of bacteria. It can be done. I mean, the genetic code sort of structure and the sort of tRNAs which decode the codons, it can be quite similar between different types of bacteria. So it can be done. I would say the challenge is having efficient, maybe not design isn't the problem, but the big DNA problem. So integrating 100 kb of synthetic DNA efficiently in a
Mhm.
reasonable time scale. I think that's the challenge developing those tools. It's not a like for like. You can't just use the exact same let's say Rexer-based system is an example
Aha.
from E. coli, that won't necessarily just work right off the bat. And these different microorganisms, they have different sort of Rec-A type variants, different recombinases that work or don't work in their within those cells. So these are some different things, and others have researched sort of adapting and discovering new recombinating tools, which I think is quite useful.
Aha.
There's some work done by some people at the Church Lab where they've discovered new recombinating-based tools that can be ported to different microorganisms, and that's quite useful in terms of providing tools to integrate at least at a small scale synthetic DNA.
Got it. Got it. That's very useful because I want to see more recorded one in other bacteria. That's
Yeah, I think it'll happen. It's just the beginning of this wave of synthetic genomics, right? And it'll be interesting to see how useful these organisms can be, because ultimately the different organisms will have different applications
yeah.
in different environments. So that it's going to be an interesting future. Yeah,
Okay, I now see the 13 uh after 11 Central Time, so probably we need to close, but we could chat
that sounds
afterward.
good.
Okay,
Yeah,
so I would say thank you all for joining and staying today. We'll meet again on August 7, Thursday, the same time, the same Zoom link, we'll have Professor Mark Stinzinski at Georgia Tech and Professor Niels Arboreste at the University of Florida. As usual, the follow informal chat will occur without recording. Please stay here if you are interested in chatting with us. I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks. I stop recording. Give me 1 second. Stop recording.